Hello, Chem 345. Today we'll be talking about the Aspirin Analysis Lab. The three learning objectives for today are to extract and quantify the active ingredient in aspirin, to explain the role of pain relievers in the body, and to describe the hydrolysis of aspirin in terms of pKa differences. Salicylic acid was first isolated in 1829, then synthesized on a large scale later in the 19th century. It has many uses, including pain reliever, fever reducer, and anti inflammatory. However, it was bitter tasting and had many side effects, including mouth irritation and gastric bleeding. Aspirin, which is acetyl salicylic acid, was a much better derivative of salicylic acid that was first put on the market in 1899. Aspirin is in the same class of drugs as ibuprofen and used to relieve pain, headaches, and fevers. In the human body, there is an enzyme called cyclooxygenase, which is responsible for producing molecules that cause inflammation and pain. These molecules are called prostaglandins, and a specific one is thromboxane A2. Thromboxane A2 is responsible for aggregating blood platelets, which is a key part in blood clotting that causes heart attacks and strokes. Because cyclooxygenase is inhibited, it can no longer produce prostaglandins like thromboxane A2. Therefore, not as many platelets aggregate and there is less blood clotting. So that's how aspirin can reduce the rate of heart attacks. Aspirin has an ester functional group. Esters consist of a carbonyl group bound to one oxygen and one carbon atom. Generally, esters are smelly and often used as a scent for perfumes and such. This molecule here, the methyl ester of salicylic acid, has the smell of wintergreen. The way aspirin and other esters are modified is by acid or base catalyzed reactions, and the best way to understand this is through pKa. Another common pain reliever is acetaminophen, otherwise known as Tylenol. Acetaminophen contains an amide and phenol functional groups, whereas aspirin has an ester and a carboxylic acid. The difference in functional group changes the way each drug is digested or metabolized. Acetaminophen inhibits the same enzyme, cyclooxygenase, by chemically reducing it in the brain, whereas aspirin inhibits cyclooxygenase in the bloodstream via a different mechanism. Organic acid-base chemistry is described by pKa. pKa is the pH when half of the protons in the sample is dissociated. The Henderson-Hasselbalch equation is used to relate pKa, pH, and concentration of a given buffer. This equation should look familiar because you used it in the acid-base worksheet last week. Another way to think about pKa is in relation to compounds' conjugate base. Does a negative charge stabilize or destabilize the compound? The lower the pKa value, the more stable its conjugate base will be. To understand this concept, compare an aliphatic alcohol and phenol, the aromatic alcohol functional group. Phenol is found in salicylic acid, and its conjugate base, phenoxide, is stabilized by the delocalization of the negative charge onto the sp2 hybridized carbons in the aromatic ring. This effect makes the proton more acidic by 5 pKa units. However, this acidic proton makes the esterification reaction much slower and unfavorable. This reaction is not as simple as combining salicylic acid and acetic acid. The first step of this reaction is to protonate the carbonyl oxygen underneath acidic conditions. Then the lone pair on the alcohol will attack the positively charged carbonyl carbon to form a tetrahedral intermediate. This intermediate will then undergo a proton transfer from the alcohol to the carboxylic acid, forming an oxonium ion. It is important to compare pKa here. The pKa of a carboxylic acid, which is about 5, is much lower than the pKa of water, which is about 15. Therefore, the equilibrium lies strongly on the reactant side for this particular reaction. Going back to the reaction, water would leave and the carbonyl would reform, resulting in an ester functional group. Water would leave and the carbonyl would reform, giving an ester functional group. For this lab, we won't be synthesizing aspirin. Instead, we will be extracting the active ingredient in aspirin. The active ingredient will be extracted using ethanol. Ethanol is a good solvent because it has a much lower dielectric constant. In broad terms, dielectric constant is a good measure of solvent polarity. So, the higher the dielectric constant, the more polar the solvent. As you can see here, water has a dielectric constant of 80. Ethanol is a good solvent for this reaction because the carbon chain solvates the aromatic ring in aspirin. However, to be sure you won't lose any product, I want to show you how to correctly use a mortar and pestle. So, place all of the aspirin in the mortar and then put the pestle inside and just gently rotate around. Don't whack or vigorously stir. And when it starts to get quite powdery, add the 20 mils of ethanol and continue gently grinding until mixture becomes a fine paste. Once you've extracted aspirin, you can test to see if it is expired or not. In the introduction, we talked about the side effects of salicylic acid, so it is useful to test how much aspirin has converted to salicylic acid. Any moisture present in the aspirin bottle will hydrolyze the ester to form salicylic acid. To test to see if aspirin is spoiled, you will use ferric chloride. Ferric chloride is a color indicator because the iron will form a purple complex when phenolic compounds are present. So, if your aspirin is spoiled, it will contain salicylic acid, and salicylic acid is a phenol-containing compound, so it will form a purple complex in the presence of ferric chloride. Please remember that ferric chloride will stain anything and everything. Remember to wear a lab coat for this experiment, or your shirt could potentially be stained brown forever. Most of the mass in an aspirin pill is not aspirin. It is a filler that acts as an excipient. An excipient is just a filler. It acts as a vehicle to deliver the active drug molecule to the body. Typically, excipients like starch are used to control the release of the drug as it is digested. You will be able to calculate the amount of aspirin in each tablet by weighing the mass of tablets and the mass of excipients.